In my previous lectures, I have talked about different cell organelles like mitochondria, endoplasmic reticulum. Now, one of the important cell organelle is chloroplast. It is present in eukaryotic cells, but you will see mostly it in green plants. Those plants which are green. In case of animals, it is absent. It is not present in animal cells. However, some protists also possess chloroplasts. Chloroplasts were first seen by Leeuwenhoek. Leeuwenhoek was a zoologist. He was related more with zoology, concerned more with zoology. But his main achievement is you are seeing that a microscope in your lab, compound microscope, also known as light microscope, that was designed by Leeuwenhoek. And he also studied chloroplasts. Chloroplasts are called green plastids. Green plastids. There are two other types of plastids, also leucoplasts, which are colorless plastids, and chromoplastids, which have color, but other than green, like these can be red, these can be blue, etc., without greenness. Chloroplasts, you have to take a leaf and then you can study it. As per shape is concerned, you will see that shape of chloroplast is different, like it can be round, it can be elliptical, or you call it oval, it can be discoid. Even some chloroplasts are cup-shaped. There are different shapes of chloroplasts as per the need. When we study chloroplast under a microscope, mostly electron microscope is used here, you will see that each chloroplast has a membrane or called envelope. And when you will see this envelope deeply, you will see it is actually two-leaved. So this is envelope or this is membrane. You can call it envelope, or you can also call it membrane. These are now two membranes, outer membrane and inner membrane. Both the membranes are of same structure as of this plasma membrane. Both are made up of lipids and proteins, or we say lipoproteinaceous in nature. That means both have lipids in them, both have proteins in them. But there is one difference between outer and inner. This is outer membrane and this is inner membrane. The outer membrane is permeable. Outer membrane is permeable. And inner membrane, it is selectively permeable. That means permeable means anything can pass through it, out or in, whereas inner selectively permeable, it selects the material. But how? The outer membrane contains small pores in it, which are called porins. These allow the material to pass through it, any type of material. These do not select the material. But as for inner membrane, this membrane, these have some proteins. These are called permeases. Proteins in it, and these proteins allow the material to pass through it under active transport, and these have the power of selection. That means if salt particle is needed, It will help transport. If it is not needed, it will not help. So material can pass from outside to inside through selective process, through permeases, and there is no binding in the outer membrane. Material can pass through it anytime. It does not select, it does not prevent, it allows it. This is the main difference between outer membrane and inner membrane. Once again, I will repeat. Outer membrane has small pores called porins which allow the material to pass through it. It does not select, anything can pass. Whereas inner membrane has small proteins called permeases which select the material and then pass through it. Usually there is active transport in it. Now, the two membranes, they have a space called intermembrane space. This is intermembrane space, this intermembrane space. I have written in short IMS, intermembrane space. Now, inside you will see fluid, colorless fluid, which is called stroma. We can also call it matrix. It is colorless fluid, usually it is called stroma. This stroma contains, for example, you will see circular DNA here in the stroma, same DNA as of prokaryotic cells. You can see ribosomes, 70S type. There are RNA molecules. RNA are of all the three types, that is transfer RNA, messenger RNA, and ribosomal RNA. All the three types: transfer RNA, messenger RNA, RNA. I have written tRNA, mRNA, rRNA. T means transfer RNA, M messenger RNA or RNA. These RNAs are important along with amino acid molecules, amino acid A. And when these are present, DNA is present, ribosomes are present, RNA is present, amino acid present, proteins can be formed. So, I have given you something about this mitochondria. Mitochondria also have all the structure. That means there is one resemblance between mitochondria and chloroplast. That is, both can help in manufacture of proteins with the help of circular DNA, ribosomes, amino acids, and transfer RNA. Other cell organelles cannot. Cell can itself synthesize proteins, and other than cell, chloroplasts can synthesize, and mitochondria can synthesize. So that was resemblance between mitochondria and chloroplast. Now, the main function of chloroplast is photosynthesis. That means formation of food. For that purpose, there are small disc-like structures, small pellet-like structures in heaps, just as this. These pellet-like structures, I will draw one pellet here. This pellet, this is called thylakoid. This is called thylakoid. Or you can also call it lamella. One is lamella. This lamella bears a space. This space. This space is called a loculus. Cavity-like structure. Space called a loculus. The loculus, it contains chlorophyll, a pigment which can help in photosynthesis. Chlorophyll, that is present in loculus, chlorophyll. And so thylakoid, actually it has been seen, if the chloroplast is very young, the thylakoids are actually parts of the inner membrane, but then they separate from it, like they are cut from it and fall in the stroma, and here they form a heap, a pile, which is called granum. A granum may contain about 60 to 100 these thylakoids on heaps. You will see numerous these grana here. Numerous grana will be present, and these grana are interconnected. The connection is called a thread. Is a thread, also known as stroma lemon. Thread or stroma lemon. Numerous grana, many grana, 100 up to 50 grana can be present in a cell. It depends upon the size of the leaf, size of the chloroplast. Now, the main function of the chloroplast is photosynthesis. This is the main function. The main function is photosynthesis. Main function is photosynthesis, that is, chloroplasts help in the formation of food for a plant. Other function is it can also form proteins, but the primary function is that with the help of chloroplasts, it traps sunlight, water is available. There are two reactions which occur here. In chloroplast, it is called a light reaction, which occurs in D. And in stroma, it occurs as dark reaction. In light reaction, the chlorophyll traps sunlight, and with the help of sunlight, it breaks water into H2 plus O. It is actually H positive is formed here. Then in the night, this energy is utilized in breaking water into H2 and O. It is also called photolysis of water. This occurs in light. That is called light reaction. Then when there is night, this stroma absorbs carbon dioxide. This is called a fixation of carbon dioxide. It occurs in night, and this carbon dioxide it then combines with hydrogen. For example, here hydrogen was produced. This hydrogen moves to stroma. Here carbon dioxide comes. Carbon dioxide and hydrogen, they fuse and they join and form C6H12O6. These are the two phases of photosynthesis. It is a separate question, but this whole process, that is photolysis of water, light reaction as well as dark reaction, it occurs both in chlorophyll as well as in stroma. In chlorophyll as light reaction, as in stroma as dark reaction. Okay, this was structure of chloroplast.